Hello, Bio 5B students. My name is OG, and I'm going to be your SI today, taking you guys in a lesson through your minds, the nervous system to be exact. I'm actually a neuroscience major, so this lesson is going to be particularly a little bit more fun and interesting for me. But as a quick preface, there's going to be a couple of times throughout the video where I'm going to ask you guys some questions, and I'll take a second or two to pause. I really want you guys to take that time to pause the video and find an answer for yourselves and see if it matches what I have down. But with that being said, let's go ahead and get started. So we're going to go ahead and get started with the basics, neural morphology. So like every single cell in your body, there's a nucleus, mitochondria, organelles. They have to be put somewhere. And for neurons, they're no different. They have the same, except they're put in here, the cell body. And this cell body, otherwise known as the soma, is going to have a bunch of extensions coming from it, or like otherwise known as processes. These processes are specialized tissue that are made for receiving input from other neurons. Do you guys know what these extensions are called? Hopefully you're able to find out for yourselves, but these are called dendrites. So dendrites are specialized nerve tissue for receiving input, and they're going to take this chemical input, convert it to electrical input, and then send that signal down to the cell body, and it's going to kind of all come together, integrate right here. And so this part of the axon, what is this part called? Hopefully able to find for yourself that this is called the axon hillock. And so essentially, if enough excitatory stimulus is received at the axon hillock, it'll generate an action potential. And that action potential will propagate throughout the entire axon and then will reach the next cell. So right now, this is the presynaptic cell. And right now, this is the postsynaptic cell. Right here, where the presynaptic cell meets the postsynaptic cell, and there's a little junction. And what is this junction called? Hopefully you're able to find out that this junction is called the synapse. So makes sense, right? What's before the synapse is the presynaptic cell. What comes after is the postsynaptic cell. And it's not like any one cell is always the presynaptic or the postsynaptic. It's always in reference to whichever synapse you're talking about. So now we're going to go a little bit more. Also, one thing I wanted to note is that signals only go in one direction. So if there's a signal coming from here, it won't all of a sudden come to the middle and then just start going backwards. They only go one way. And so with that being said, I'm going to go a little bit more into actually how the signal is being passed through membrane potentials. So membrane potentials. Membrane potentials are, what is a membrane potential actually? Hopefully you're able to find out for yourselves that membrane potentials are a charge difference across a membrane. So it's where you accumulate enough charges on one side versus another side. And I kind of have that shown right here in this little diagram, zoomed in diagram of an axon. And so as you can see, there's a lot more negative charges on the inside and there's a lot more positive charge on the outside. And note that this is always in reference to each other, but we'll go into that later. And this membrane potential is sort of like a polarized form at resting potential, and it is maintained by a sodium potassium pump. Do you guys remember whether there's more sodium on the outside or more sodium on the inside, and vice versa for potassium? Hopefully you're able to remember that there's a lot more sodium on the outside and there's a lot more potassium on the inside. That's the way that the sodium potassium pump pumps these relative ions. And the little mnemonic that I used to help remember this is this little thing called a salty banana. It's kind of funny, it's kind of silly, but it helps me remember that salt, which has a plenty of sodium ions, and potassium, which or bananas, which have plenty of potassium in it. So I'll put bananas and a bunch of potassium. And as you can see, they have highlighted here 3 and 2. That's because each single round of the sodium potassium is pumping out. It's pumping out 3 sodiums outside and 2 potassiums inside. And so I'll kind of draw that in over here in our little diagram. We have 3 sodiums on the outside and 2 potassium on the inside. Obviously, there's a general accumulation of it, so it's not literally just 3 ions and 2 ions. It builds up, but hopefully this can help explain why the inside is negative, even though there are positive potassium charges on the inside. It gets a little bit confusing about this, and I know I definitely had a question about it when I was taking this class, but think about it that the inside is a lot less positive than the outside. Or outside, there's a lot more positive charges on the outside than there are on the inside. So relative to the inside, outside is more positive, and relative to the outside, inside is a lot more negative. Hopefully that made a little bit of sense, but like I said, if you guys have any questions, please do look up more YouTube videos or take reread the book or rewatch through the lectures, because this can definitely get a little bit complicated. So I'm going to kind of go through a really brief rundown of how this actual electrical signal is being passed down. This it can get really, really complicated, but put in simple terms, it's essentially a cascade of an opening of sodium channel. So that's the opening of a sodium channel is going to lead to depolarization of the membrane. So we have a signal that's had already been coming from, let's say, the soma, and reaches this part of the axon. So at this part of the axon, there are sodium channels that are now being opened up. This is the first step. So sodium channels are being opened up and they're being released inside the neuron. And so when the sodium comes in, you can notice that there is a change in membrane potential. Right here, it is no longer negative on the inside. And so because of that change in membrane potential, if you guys remember, there are ion channels that are specific for noticing that. I'm going to put some of them right here, and right here. They're all over the place. So do you guys remember what are these ions, specifically ion channels that are meant for noticing changes in voltage? Hopefully you remember, these are the voltage gated ion channels. So these voltage gated ion channels will notice the difference in voltage and will then open up itself to let more sodium flow through. And that cascade is going to continuously happen down the axon, and it's only going to go in one direction. And later, as you can see, potassium is then let back out to kind of restore the charge differential where it's positive on the outside, negative on the inside. And this whole cascade kind of carries on down through the axon. This is the electrical component of signal transduction down an axon. All right, so next thing we're going to be covering is, so kind of zoomed back out into the whole neuron. Next thing we're going to be covering is right here with the synapse and what's actually going on with the synapse. So if you don't mind me, I'm gonna just blow that up. And so right here, this is a close-up version of the synapse. And so essentially, as an action potential is being propagated along and it gets to the nerve terminal, at this point you have several factors that are several players that are playing a role here. So you have neurotransmitters, and these neurotransmitters are being packaged up into these little balls called vesicles. And when the signal gets to the nerve terminal, it releases these neurotransmitters into this little area right here. This area is called the synaptic cleft. And so this is the presynaptic terminal, and this is the postsynaptic cell. And so after it gets released into the synaptic cleft, it is then received by these receptors right here. And so these receptors are actually specialized for receiving neurotransmitters and then leading to a cellular response. What those cellular responses may be is a little bit outside the scope of the class, but just know that this is how it receives the chemical input from another cell. And the next important thing that you guys need to know is how are these neurotransmitters degraded or inactivated? So one of the ways, especially with acetylcholine, is there are these degrading enzymes that are often floating around in the synaptic cleft. And so these degrading enzymes will then take the neurotransmitters that didn't get received and degrade them as instantaneously. Some of them are near catalytic perfection, which means they work really, really quickly. Another method of getting rid of the neurotransmitters is these reuptake transporters. So neurotransmitters in the synaptic cleft can also be taken back into the presynaptic membrane through these transporters. After they're back inside, they can then be degraded by metabolizing enzymes or taken back in and stored in more vesicles for later release. So that's kind of a very basic rundown of the synapse. Obviously, like I said, there's always more to it, but this is all I'm going to cover for now. So yeah, the next part that we're going to go into is the parts of the brain. So two of the major parts of the brain is the forebrain and the hindbrain. In the forebrain, you have the telencephalon and the diencephalon, and in the hindbrain, the prominent part of it is the cerebellum. However, my question for you guys is, do we or do we not have a midbrain? So hopefully you guys were able to find out for yourselves that we indeed do have a midbrain. It was not evolved away. It's found right here at the top of the brainstem. And I'll label it for you right here, midbrain. And yeah, you don't really need to know what's inside the midbrain, but just know that we do have it. Next, I'm going to go into are the four major lobes of the cortex. So we have the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. There was a lot of information that she went over in class about it that I'm not going to have the time to go through right now, but I'm going to kind of give you a very shortened OG version of what are the major functions of each lobe. So with the parietal lobe, you have sensory integration. So many of your senses are integrated right here. Talking about senses like taste and touch and all that. For occipital lobe, over here you have visual perception and processing. For temporal lobe, you have hearing. So as you can see, the temporal lobe is on the sides next to your ear. Makes sense. So this is where this is your hearing center. And also with hearing comes language perceptions, because language are things that you hear. And so that's actually here in the Wernicke's area, but I actually don't have that written down here for you guys, but do please take the time to go into that. And last, but certainly not least, is the frontal lobe. And the frontal lobe, I personally like to call him the boss, because him or her, I like to call the frontal lobe the boss, because the frontal lobe is basically the part of your brain right here in the front that makes all of your executive decisions, all of your decision making, attention, arousal. It's kind of all controlled by the frontal lobe. So this is what tells the rest of your brain essentially what to do. So the last thing I'm going to go over with you guys is plasticity. So there's two types that you guys need to know for this class. There's neuroplasticity and synaptic plasticity. And so with neuroplasticity, the definition of that is just increasing or decreasing synaptic connections in response to usage. So in this little diagram you have here, you have this neuron that's being synapsed by two different neurons, neuron 1 and neuron 2. And so as neuron 1 is being used a lot more than neuron 2, it's going to lead to an increase in connections of neuron 1 and a decrease in synaptic connections in neuron 2. And this is what you call synaptic plasticity. It's associated with just learning new or refining skills, like different types of motor skills. And lastly is synaptic plasticity. So for synaptic plasticity, it requires repetitive, excitatory stimulation. So back-to-back stimulation that ends with the release of an excitatory neurotransmitter, such as glutamate. And when you have these repetitive excitatory stimulations, it's going to lead to long-term potentiation, which essentially leaves the postsynaptic cell much, much more sensitized to neurotransmitters. And this sensitization happens through an increase in receptors. So when you have more and more receptors that are found on the postsynaptic neuron, it basically becomes much more sensitive and much more responsive to input coming in from the presynaptic neuron. So that's kind of a quick sort of differential between neuroplasticity and synaptic plasticity. Again, neuroplasticity, there are more synaptic connections, and synaptic plasticity is when you strengthen the connections you already have. But yeah. So hopefully you guys enjoyed this video, this review on the nervous system. There's obviously a lot more to it than I was able to get to in the video. So I strongly recommend to rewatch the lectures, look inside the book, and just maybe even look up other YouTube videos for more information. If you guys have any questions, please feel free to comment down below. We'll get back to you as quickly as we can. But yeah, that's all from me. Hopefully you guys have a wonderful rest of your week, and I'll see you guys around. Later.